Hello, I am Mr. Yash Suryawanshi from final year Bachelor of Pharmacy from Shivaji Rao S. Zondoi College of Pharmacy. My topic name is Gold Nanoparticles Synthesis and Application in Cancer Diagnosis and Therapy, and this topic is guided by Dhanashri Kathole ma'am. Metal nanoparticles are widely employed in biomedical applications because of their high thermal stability and low size to volume ratio. Due to their simplicity in synthesis, stability and functionalization, low toxicity, and simplicity in detection, gold nanoparticles are natural choice for biomedical applications. Synthesis of gold nanoparticle has been carried out using a variety of chemical techniques over last few decades, but more recently emphasis has been drawn to more environmentally friendly green methods. Development of new gold nanoparticle synthesis technique is the main emphasis of this review. Gold is inert and universally recognized as biocompatible. Until the recent past, it was only known as the metal. With the arrival of nanotechnology, the discovery of nanoparticles, and exploration of physicochemical properties of gold make it a supreme material for progress fields. The term nanotechnology refers to the development and the use of parts that are produced at nanoscales of up to 10 to 1000 nanometer in size. Nanotechnology includes study of molecular and submolecular structural characteristics or nanostructures as well as their electrical, optical and magnetic characteristics. Nanotechnology is now an interdisciplinary topic that unites engineering, biology, chemistry and physics. In recent years, there has been a lot of study on and interest in the field of nanotechnology. The fast advancement of nanotechnology has led to remarkable expansion in production and application of nanoparticles. One of the most significant nanoparticles is gold nanoparticles, and owing to its inert, biocompatible, and particularly low toxicity properties, they have been employed extensively for both medical and non medical applications as excellent materials. This world has entered a new period known as the era of nanotechnology, thanks to the use of nanomaterials in many industries such as oil and gas, cosmetics, and nanomedicines. Synthesis of gold nanoparticle. First method is Turkevich method. The resulting gold nanoparticles are between 1 and 2 nanometers in size. The primary idea behind this method is to use reducing agents such as amino acids, ascorbic acid, UV radiation or citrate to convert gold ions Au3+ into gold atoms Au0. A variety of capping and stabilizing chemicals are used to stabilize gold nanoparticles. This approach initially has a limiting number of applications due to the variety of gold nanoparticles that could be produced using this process. Burst method. This method uses a two-phase process and organic solvents to create gold nanoparticles with a size range of 1 to 5 to 5 to 2 nanometer. The process includes moving gold salt from its aqueous solution to an organic solvent like toluene using a phase transfer agent like tetraacetyl ammonium bromide. The gold is subsequently reduced using an alkane ethyl and a reducing agent such as sodium borohydrate. The stabilization of gold nanoparticles is carried out by the alkane ethyl. Seed mediated growth method. The process of seed mediated development is one of that most frequently used to create rod shaped gold nanoparticles. The underlying idea behind this procedure calls for first manufacturing seed particles by reducing gold salts. Reducing agents like NaBH4 are used during this process. The seed particles are then transferred to a metal salt and a mild reducing agent such as ascorbic acid to stop further enucleation and hasten the synthesis of rod-shaped gold nanoparticles. Other method is digestive ripening. In the presence of too many ligands, digestive ripening agents, digestive ripening is thought to be a practical way to create monodispersed gold nanoparticles. According to the figure B, the fundamental procedure is heating a colloidal solution at a high temperature, that is about 138 degrees Celsius for 2 minutes, and then heating it at 110 degrees Celsius for 5 hours. The primary factor affecting the size distribution of the gold colloids is temperature. Various types of gold nanoparticles can be synthesized, such as gold nanorods, gold nanocages, and gold nanoclusters. First, gold nanorods. According to the research, gold nanorods are modified with the RLT polypeptide, which binds to low density lipoprotein binding domain have a fairly noticeable inhibitory impact on gastric cancer cells. Additionally, they showed improved biosafety in vivo as compared to the free medication doxorubicin and considerable anti-tumor activity in the treatment of malignancies. This gold nanorod can promote mitochondrial malfunction, lessen the capacity of mitochondrial membrane to activate cell apoptosis, increase the formation of singlet oxygen, and decrease the ability of gold nanorod to bind serum proteins. Second types are gold nanocages. One variety of gold nanoparticles is called as gold nanocage, which has a hollow cage-like structure. They are more drug-loaded than the other two nanoparticles because of their high specific surface area, excellent surface modifying area, and high specific surface area. The combination of rubefacin A, hyaluronic acid, anti-CBC antibody, and gold nanocages inhibit pancreatic cancer. Additionally, gold nanocages have been capacity to image in several modes including magnetic resonance imaging and near infrared fluorescence, which can be identify pancreatic cancer at early stage. A compound was created by combining PDL1 antibody and a TGF inhibitor and gold nanocages which can target colon cancer cells with precision and gather in tumors. Third types are gold nanoclusters. When compared to gold nanorods and nanoclusters, gold nanoclusters have significantly lower cytotoxicity and superior red fluorescence properties, which enable them to successfully avoid autofluorescence background in vivo. Gold nanoclusters are extremely small particles made up of few to few hundred gold atoms, and they are composed of several to a few hundred gold atoms. Gold nanoclusters coated with folic acid linked silica have also been created, and they efficiently target stomach cancer cells and have a good red fluorescence optical qualities. They can be employed for optical and CT dual mode imaging of gastric cancer and have a potential in applications of in vivo early detection of gastric cancer. Application of gold nanoparticles in diagnosis and therapy of cancer. The gold nanoparticles have low in vivo toxicity and are non-immunogenic and are stable. Additionally, they can accumulate in tumor areas, making them appealing for imaging diagnosis. The use of conventional CT contrast agents is constrained by their small molecular size, short circular duration, and adverse side effects like nausea and itchiness. Due to their high X-ray attenuation coefficient and biocompatibility, gold nanoparticles are a potential CT contrast agent. By targeting mouse colon cancer cells and serving as a CT contrast agent, a monoclonal antibody against HSP70 conjugated to gold nanoparticles demonstrated outside imaging capabilities in spectral CT and excellent sensitivity for identification of even single cells. These are some examples of gold nanoparticles which are used for detection of various types of cancers. Gold nanoparticles have a significant impact on management of gastrointestinal malignancies. A novel cancer therapy called photothermal therapy can target tumor tissue and heat it while sparing healthy tissues from harm. The plasmonic nanoparticles are NRI radiated and then transported to tumor cells or tissues, where the absorbed light is transformed into heat and irreparably harming the neighboring diseased tissue. Many different nanoparticles are employed in PTT, but gold nanoparticles in particular can passively accumulate in tumor tissues. Additionally, their structural makeup may be changed to the best absorb an IR light from 650 to 1350 nanometer, which has become a significant therapeutic platform for photothermal treatment. With the use of photosensitizing medications and laser activation, photodynamic therapy (PDT) is a revolutionary approach for treating neoplastic disorders. When the photosensitizers are applied to tumor cells or tissues and exposed to a particular laser wavelength, a highly reactive oxygen species is created that slows down the development of tumor. Indocyanine green (ICG), a tricarbocyanine dye that has been given FDA approval as a possible near-infrared photosensitizer for clinical imaging and diagnostic, is the most widely used photosensitizer. However, due to its low stability and quick blood elimination, ICG cannot be used for photodynamic treatment or fluorescence imaging. Therefore, it is a good idea to create a new cancer therapy that combines PTT and PDT to treat gastrointestinal cancer. In order to target the endoplasmic reticulum, hollow gold nanospheres were first coated with endocyanin green and then modified with FAL polypeptide. This created a PDT composite nanosystem. When exposed to NRI radiation at 808 nanometer, there was a simultaneous rise in temperature and ROS production which caused ER stress and boosted the immunological response. ICG molecules were conjugated to gold nanoparticles using a linker made up of branched polyethylene glycol, which has molecular weight of 10 kilodalton. By combining PTT with PDT treatment, this innovative nanosystem can prevent tumor development and metastasis. Advantages and disadvantages of gold nanoparticles. Advantages: gold nanoparticles size can be manipulated easily for active and passive drug targeting. Drug can be incorporated without any chemical reaction. Site-specific drug targeting can be done by attaching targeting ligand to surface of gold nanoparticles. Administration through various routes like oral, nasal, and parenteral is possible. Gold nanoparticles increase bioavailability. Disadvantages: it may alter physical properties and can cause particle-particle aggregation. They are very reactive in cellular environment. Small particle size are responsible for limited drug loading. It is more expensive and short shelf life. Routes of administration of gold nanoparticles. Oral administration. Advantage: it is non-invasive means of nanoparticle delivery. Disadvantages: first pass metabolism occur, hepatotoxicity occurs, potential for translocation into systemic circulation, and it requires intact intestinal mucosa for nanoparticle uptake. Second route is transdermal route. It has advantages like it is a non-invasive means of nanoparticle delivery. It has large surface area and it gives local action. Disadvantages: it can cause local irritation, potential for translocation into systemic circulation. Third route is pulmonary route. It has advantages as it is non-invasive means of nanoparticle delivery, large surface area, it gives local action and avoid first pass metabolism in liver. Disadvantages: it can cause local toxicity, potential for translocation into systemic circulation. Fourth route is intravenous route. It has advantages of systemic delivery of nanoparticle, systemic action, and disadvantages is it can cause first pass metabolism in liver, potentially hepatotoxic, and can cause systemic toxicity. These are recent formulations of gold nanoparticles. In this review, we have discussed various methods for synthesis of gold nanoparticles, types of gold nanoparticles, and applications of gold nanoparticles in cancer diagnosis and therapy. Gold nanoparticles are used to identify and treat gastrointestinal cancer. Thus, gold nanoparticles are a potential imaging, diagnostic, and therapeutic tool for gastrointestinal malignancies. These are the references I used to make this review. I would like to thank Dr. Shivajirao S. Zonte, President, and Dr. Geeta Khare Ma'am, Secretary. And thank you. Hope you like this video. For more informative videos, subscribe to our YouTube channel.